our next speaker is Lee Sweeney, who's here from the University of Pennsylvania, and he is the director of our Wellstone Center. And he's going to be talking about from targets to clinical trials in muscle disease. Yeah, this one. And there's this corner if you want that corner, where it comes out.
Sorry, couldn't find the right button. Yeah, I'm going to tell you a little bit about a subset of the work that's going on that could benefit the Becker population, either in the near term or in the years to come. Different therapies that are being developed that would be useful. As Beth said, I'm at the University of Pennsylvania, and I work with a distributed group of investigators across the country, and our goal is to try to bring new therapies to both Duchenne and Becker muscular dystrophies. You're going to hear from two of my collaborators afterwards, and one of which, Ron Victor, will tell you about a trial that has begun with Becker patients and hopefully will be extended. So in Becker muscular dystrophy, there are a number of different things that go wrong because the dystrophin protein that's being expressed is either unstable or only partially functional. And one can try to do some things that would mitigate some of the problems that are caused by this. And the first one I'm going to talk a good bit about is this idea of increasing muscle mass and regeneration as a way to sort of improve the strength of the muscle and the quality of the muscle. Another way, which is moving toward the clinic in a variety of ways, is trying to decrease inflammation and fibrosis, which you've probably already heard from the other speakers. It gets in the way of the muscle repairing itself. And then Ron's going to talk to you about an approach to try to correct the blood flow regulation that is disrupted in the majority of Becker patients, not all. And then there are also perturbations in calcium handling in both the skeletal and the cardiac muscle, and if you could try to normalize that, that would go a long way to slowing the progression of the disease, and maybe profoundly so in the heart. So as I said, I'm going to focus my talk on this first topic of increasing muscle mass and regeneration. So all of us, when we damage our skeletal muscles, go through the same cycle of injury and repair. The problem in Becker muscular dystrophy is that the threshold for injury is much lower, and so it takes much less activity of the muscle to lead to the equivalent amount of damage that would normally occur. And because of this, you have this ongoing cycle of muscle injury, which then causes breakdown of the muscle. That you have an inflammation because the inflammatory cells have to come in to sort of deal with the damage, clear out the debris, and set up an environment where the repair can occur. So the signals that come out of the inflammation are actually important for triggering many of the phases of regeneration. You have within the muscle cells that are closely opposed to the muscle membrane called satellite cells, and these are essentially muscle stem cells that are there to repair the muscle whenever it's injured. And so during the inflammation phase, these satellite cells are activated to begin to repair the muscle. They are cells that are normally not doing anything, but once they're signaled to repair, they start dividing, and then they can fuse into the muscle membrane, or they can actually form new muscle fibers altogether. So this remodeling and repair involves a good bit of remodeling of the extracellular matrix as well as blood vessels to serve the muscle before you can rebuild the damaged muscle. So as I said, the problem in Becker muscular dystrophy is this happens at such a high rate that in fact, in many cases, the inflammation remains elevated and actually begins to interfere with the regeneration. And so the result of that is that some of this pro-inflammatory cytokine environment doesn't really go down as it should, and so you end up having remodeling that is not competent. You end up having matrix that becomes fibrotic, and the satellite cells don't get properly activated, and so slowly you can replace the muscle with fibrosis, and then probably later with a combination of fat and fibrosis. So one way to sort of combat that is this idea of signaling better repair mechanisms, and the mechanism that it's actually in clinical trials now, and I'll tell you a little bit more about the details of the trial, and that's using a virus to deliver follistatin, and I'll tell you why follistatin. You also are going to hear in 2 talks from now about another mechanism, PD5 inhibition, which we think would be limit the damage to some extent for skeletal and cardiac muscle, and may also have some consequences that would aid in the repair as well. But I'm going to talk about myostatin inhibition. So follistatin is something that can carry out at least a form of myostatin inhibition. Now, what is myostatin inhibition all about? Well, the idea is you want to have these satellite cells that I talked about, you want them to be able to better repair the muscle. So you want them to be able to get signaled and divide and not be interfered with and go ahead and repair the muscle. So that normally is under control of a number of growth factors. So you have one type of growth factor that can activate the satellite cells because it's released when the injury occurs. And then you have several other growth factors, perhaps the most important of which is IGF-1, that can stimulate these satellite cells to divide or proliferate, and then eventually to differentiate and repair the muscle. But you also have a set of growth factors that are, in the case of skeletal muscle, are sort of anti-growth factors, if you like, the leading one being myostatin, but to some extent activin A and TGF-beta. These are actually all members of the TGF-beta family, members of protein, the family of proteins, of growth factors, and all of these can inhibit the proliferation of these satellite cells. So in essence, they work against IGF-1. And normal muscle growth and repair is sort of a balance that's set by the growth factors such as IGF-1 and antagonized by myostatin and TGF-beta. Now, myostatin was not even known when I was in school. Myostatin was actually not discovered until the mid-1990s when Se-Jin Lee was at Johns Hopkins was conducting a series of genetic experiments where one by one he was knocking out TGF-beta family members in mice and then asking what happened, because many of them had unknown actions. One of the ones that had unknown actions was one called GDF8, which he renamed myostatin. And when he knocked it out, it gave this amazing phenotype in mice. The mice developed these gigantic muscles, essentially muscles that were double the size of normal mice. So Se-Jin immediately recognized the potential importance of this because what he had found was a negative regulator of muscle growth. And what he saw in his later investigations was that it is a member of the TGF-beta family, and it signals through the same receptors as many of the activin members of the TGF-beta family. In particular, it can signal through this, what's called the activin 2B receptor, and it leads to a signal cascade that leads to gene expression that actually inhibits the growth of muscle, skeletal muscle. Now, what's interesting about myostatin is it's relatively selective for skeletal muscle, so you could probably interfere with myostatin action and not have big effects on other tissues in the body, so a relatively targeted therapeutic approach. And these are ways that what people have shown, and Se-Jin and other investigators have shown, that you actually can block myostatin signaling actions and thereby relieve its inhibition on skeletal muscle growth and repair. You can develop an antibody against myostatin, which then takes out the myostatin. Follistatin, which I mentioned before, is in clinical trial. is a protein that circulates. It's also found in the tissues, and it binds myostatin, but it also binds other members of the activin family, so it inhibits more than just myostatin. The myostatin propeptide also is an inhibitor, so this is a piece of myostatin that can inhibit the sort of docking of the full myostatin, and therefore causes depletion from the tissue and inhibit its actions. And then a soluble version of the receptor itself, the activin 2B receptor, acts as a decoy if it's in the blood and will soak up myostatin, but it also soaks up a number of other things. So the question is, Sagen originally showed that if you knock this out in the genome, then you have these big muscles. But what happens if you just inhibit myostatin after birth? Well, it turns out that's also quite effective. And as I said, a number of strategies have been developed to do this and have been shown in a number of animal models. Probably the first one that was developed was a neutralizing antibody against myostatin that was developed by Wyeth. Pfizer has purchased Wyeth in the years since, and they now are developing a very much improved antibody. The original antibody failed in clinical trials, not because myostatin was a bad target, but because the antibody they were using was not a very good drug. So they have a much better antibody that they now have started dosing patients with. It looks very promising, but it's in very early phases. The myostatin propeptide has never been put into people, but it's been very effective at inhibiting myostatin in animals. And then these last two, follistatin and the soluble version of the activin 2B receptor, as I mentioned before, are very potent inhibitors of myostatin, but they also inhibit a number of other things, which systemically can lead to complications. And for example, well, this is just showing you this profile. This is now the soluble activin 2B receptor, and it inhibits a lot of things other than just myostatin. In fact, it inhibits these activins and BMPs actually quite a bit better than it inhibits myostatin. So the problem is if you dump a lot of this receptor, this soluble version of the receptor into the blood, it's going to soak up all of these activin family members, not just myostatin, and block a lot of signaling that you might not want to block. Putting high levels of follistatin in the blood would have the same effect, and so both of these are problematic. But if you were to target it just to the muscle, then it's been shown that they really can cause much more hypertrophy in the muscle than just blocking myostatin alone. So what's been shown in animals? Well, blocking the myostatin pathway leads to much larger muscle fiber size and greater strength in normal mice. If you go into dystrophic mice, you can improve muscle regeneration, you can increase the muscle size and strength, and you can decrease fibrosis. Now this carries over into dogs. We've looked at dystrophic dogs, and again, inhibiting myostatin can improve muscle regeneration, increase the size of the muscle, the strength of the muscle, and decrease fibrosis. And then lastly, this now has been looked into some extent in non-human primates and monkeys, and again, in inhibiting the myostatin pathway will lead to bigger and stronger muscles. So this looks like a great target, and especially a great target, I would argue, for Becker muscular dystrophy. So here's just showing some MRI from the dog study that we did and collaborated with Glenn Walter, who you're going to hear from next. So this is looking at the size, at different muscle sizes in these dystrophic dogs, looking at their cross-sectional areas, looking at their muscle volumes. And this is MRI is an important tool here because we can look at the effect of myostatin inhibition in all of the muscles in terms of the impact on size and fibrosis without having to take biopsies from all the muscles, because now we can see all the muscles in a very non-invasive way and do it time and time again as the therapeutic window increases. So this is what we saw in the dystrophic dogs. So these were full-grown adult dogs. They have the subset of dogs that we used had a milder form of dystrophy compared to some, and so you might even characterize these as Becker-like subset of dogs. And so in these dystrophic dogs, we intervened at time 0 here, and then over over the next year, you see there was an increase of the muscle size. Now, normally in these dystrophic dogs, they would lose muscle over a year, but here they're increasing their muscle, especially over the first few months, as a result of this myostatin inhibition. And on average, about 20% increase in the cross-sectional area of the muscles. The same is seen if you just look at the volume of the whole anterior compartment of muscles, about a 20% increase that was maintained over that year after treatment with this myostatin inhibitor. And so this was continuous exposure to the propeptide to inhibit myostatin signaling. What we also saw was there was a decrease in fibrosis over the year in these muscles, and the muscle looked much better than muscles that did not have myostatin inhibition. There was much more successful regeneration going on, less central nucleation. Now, follistatin, as I said, is a much more potent inhibitor of these inhibitors of muscle growth, because it not only inhibits myostatin, it also inhibits activin A. And it turns out there's a fair amount of activin A in skeletal muscle that is also inhibiting muscle growth. And so this is now an experiment putting follistatin into a virus, into adeno-associated virus, and injecting it into a monkey in this, to see if you can actually, in a non-human primate, this is not a dystrophic monkey, this is just a normal monkey, but to see if you can get muscle growth using follistatin as the myostatin pathway inhibitor. And by expressing it only in the muscle, you're not going to have systemic problems like you would if you increase the levels of follistatin circulating in the blood. So this is what happened. In this monkey, one leg was left untreated, the other leg was injected with a virus that expressed this follistatin to inhibit myostatin and activin A, and you can see this giant increase in the muscle size, giant. It was like a 30% increase in the muscle size in the injected leg versus the uninjected leg. So again, this follistatin inhibits the signaling through this cascade and thereby relieves the inhibition on muscle growth. So this is now moved into the clinic for Becker muscular dystrophy, and the idea is to try to inhibit myostatin and activin A by AAV delivery of follistatin into the quadriceps muscles in Becker patients. So this is what the clinical trial is going to be like. This first bit is a small focused trial on 9 Becker muscular dystrophy patients, at least 18 years of age or older. They must have a confirmed mutation that gives rise to Becker. They're going to undergo pre- and post-gene therapy muscle biopsies. Now, they're also going to undergo MRI imaging like I showed you for the dogs, so that you get a much more detailed picture of what's going on in the muscle, but also the biopsies necessary to confirm the expression of the follistatin itself. The virus is going to be injected into both thigh muscles. Primarily, we're talking about the quadriceps here. And it's a virus called AAV1 that's coding for one of the isoforms of follistatin that's called FS344. This refers to the molecular weights, 344 kilodaltons. The outcome is going to be the 6-minute walk test. So even though it didn't say up here, obviously these are going to have to be ambulatory Becker patients because they're going to have to be able to do a 6-minute walk test. There's also going to be quantitative myometry of the knee extensors, and then the patients will be followed for 2 years to see what the lasting impact of this treatment is. So those of you who are not really familiar with AAV, the idea is you make a synthetic gene, in this case coding for the coding sequence of follistatin, and in this trial the virus is going to use the CMV promoter to drive it, and so this follistatin will be expressed wherever the virus is injected. In this case, it's only going to be injected into the quadriceps. And then the virus is packaged in such a way that it doesn't express any viral genes, just this synthetic gene to make follistatin. So the idea here is that this molecule, follistatin, inhibits both myostatin and activin A signaling within the skeletal muscle. But since it's only going to be made in the skeletal muscle, it's not going to have systemic effects. So it's not going to have side effects associated with follistatin going up in the whole body. But it also means that the the benefit is only going to be in these thigh muscles that are injected. But the main drawback of the approach is that even though it's going to make the muscles larger and stronger, almost undoubtedly based on the weight of all the animal data, it's also not going to do anything to make the muscle get less damage. It will decrease fibrosis, but the muscle's still going to be damaged, so the muscle's still going to be regenerating. And so that means over time, the follistatin expression, since it's coming from the muscle, will gradually be lost. But nevertheless, since this is Becker as opposed to DMD, the muscle turnover is much slower, and so it's probably going to be years of expression before it would be lost. And so years of benefit from the single injection of virus into these muscles. Now, the heart's not going to benefit at all from any form of myostatin inhibition, and in fact, you can argue that as you increase the load on the heart, that it's actually going to be bad for the heart. So I think no matter what sort of myostatin inhibitory trial is going on, it's really important that patients be on cardioprotective drugs of the sort that Beth talked about earlier, namely ACE inhibitors or ARBs, and beta blockers are probably a good idea to be used in these trials. There are other drugs that are being developed that could be cardioprotective, and Beth also mentioned the spironolactones. So anyway, there are a number of these pathways that I think could give rise to improvement, slowing of Becker muscular dystrophy, and even improvement in the muscle, especially from this first one, which is why I'm so keen on this idea of myostatin inhibition, because I think you can get improved function in Becker, not just slowing of disease, but overt improvement as you increase the strength of the muscles and improve the regeneration in the muscles. There are other approaches that are also moving toward the clinic. Ron Victor's going to tell you about one, but others are that there's going to be systemic myostatin inhibition via this neutralizing antibody that Pfizer is going to bring on board. So maybe a few years after this viral trial is concluded, there may be a way to get the same benefit for all the muscles of the body in a safe manner because of the fact that if this neutralizing antibody works, then you'll be able to inject it every few weeks and get growth and protection of all of the muscles. There are antifibrotic therapies. One in particular that you may be aware of is this halofuginone, which is moving toward the clinic for DMD. Anti-inflammatory drugs such as NF-kappa B inhibitors are moving to the clinic and could be useful both in DMD and in BMD. And then there are drugs that are undergoing development that might normalize these calcium leaks that occur both in the cardiac and skeletal muscle of Becker muscular dystrophy as well as Duchenne muscular dystrophy patients, and could go a long way to addressing especially the cardiac deficits. So I'm going to end with that and just put this up and say if any of you are interested and would qualify to be in this trial, then please contact Ana Marie Gomez, who's the trial coordinator, at 614-722-2715. This trial is being done by Jerry Mendel's group at Nationwide Children's Hospital in Columbus, and Ana Marie Gomez is Dr. Mendel's clinical trial coordinator. So I'll stop with that, and when we get up for the panel, I'll take any questions. Thank you.